Until you do that, I can introduce you to the audience. So
Yes,
Paolo
please.
Magni is Associate Professor of General Pathology at the University of Milan. He graduated in medicine and surgery and specialized in endocrinology at the University of Pavia. He then carried out a 3-year research experience in the USA and obtained a doctorate in endocrinological and metabolic sciences at the University of Milan. His research interests are in the cardiometabolic field, with particular regard to the pathophysiology of obesity, metabolic syndrome, and atherosclerosis, with extension to nutrition and nutraceuticals, and with particular reference to the microbiota and probiotics. He is the author of over 100 international scientific publications. So, Paolo, you can start.
Okay, thank you very much, Dimitris. So welcome to everybody. Today we talk about cell line models for atherosclerosis. Clearly, this is a very large topic, and this webinar actually, as you know, is in the list of webinars by 2 projects we share with Dimitris, Milan, and many other very important, nice colleagues, the Atheronet Construction and the Cardioscope project. Now, I don't have to tell you again that the 2 projects basically are approaching, are including a multi-omic approach and artificial intelligence strategies to address 2 the important issues. One is, generally speaking, atherosclerosis, and the other one is the acute coronary syndrome. So the goal is to have a more precise definition of the cardiovascular risk and prognosis. Now, this is the outline of the, can you hear me?
Yes, we can hear you, Paolo. Very well.
And also, you can see also the change of the slides, right?
Yes.
Great. Okay, so I will shortly comment why using cell-based models to study atherosclerosis. I will go over over some considerations about which cell types we may be considered. I will speak most of the time about 2-dimension 2D systems, which are the mostly used, but I will close with some ideas and some, let's say, more complex models, which include 3D systems, organoids, microfluidic systems. So why using cells or cell-based models? As you know from the other webinars and from your personal education and studies, you know that we start from the arterial wall, and then slowly we have some lipid deposition, fatty streaks, and then we go on with the atherosclerotic plaque, which has a specific composition, specific biology, and the endpoint can be a severe obstruction or an acute occlusive thrombus, which is a combination of atherosclerosis plus acute thrombosis. As you can see here, different events, large complexity. This is a picture taken from a recent paper by Milan and some of us. And you know that, of course, when you're talking about human cores, human populations, the complexity is really great, the complexity in the pathophysiology of atherosclerosis, and so there is an individual impact of different factors. However, it is important to kind of disentangle some of these mechanisms utilizing let's say, models, which can be, for example, animal models. You know very well that there are some, besides big animals, there are kind of these small animals. They are very much used. They have some advantages listed here: plaque accessibility, genetic manipulation, and so on. Of course, there are some disadvantages. For example, they develop plaques in areas which are not totally superimposable with those areas in humans. There are limited data on transcriptomics, for example. There is a limited genetic variability, low throughput sometimes, and some other issues. I depict here also the zebrafish, which is also a more a less expensive model system, but of course some enzyme let's say pattern may even replicate those present in humans, but still there are some limitations. Now, the final point is that can suggest us also to at least have some cell models in addition to animal ones. Are the principles of the 3 R's that you know very well: replacement, reduction, and refinement of the use of animal models. So let's see what we have. Going back to the pathophysiology of the atherosclerotic plaque, you can see here in this box here a list of cells. We start from endothelial dysfunction. Then we have monocyte adhesion, migration, and migration in underneath the intima, and which means that there is a phenotypic change, a functional change to macrophages, smooth muscle cells, and lipid accumulation. Of course, lipids are mostly coming from the liver. So which type or cell types can we consider? Okay, again, here is depicted the picture of the main cell types playing a role, at least classically, playing a role in the pathophysiology of the atherosclerotic plaque. So we have endothelial cells, which should be considered, hepatocytes, which produce a proper or an altered, in this case, lipid profile, which is associated with low density lipoprotein cholesterol increase, which also may be also oxidized. Then we have monocytes, which are adherent to the endothelial cells, and then they become macrophages, and they become then foam cells. Also, the smooth muscle cells, they usually stay here. They don't move. They just retain their contractility, but we will see they can differentiate, migrate, and generate many different phenotypes and changes. Again, this is the same picture with more details, okay, showing that, for example, a great deal of interest is regarding, among them, also the smooth muscle cells, which are actually doing many things when they go here underneath in this subintimal position. Let's start with the endothelial cells. I'm not going to a comprehensive overview because of the limitations of time, but of course, it's important to underline that Professor Kardassis is organizing and coordinating 2 papers, which I hope are going to be interesting for all of us, one on cell models and another one on animal models. While waiting for these papers, to which several of us are contributing, I just go over some examples, some ideas, giving you some, let's say, landscape of the of the cell-based models. Among the endothelial cell dysfunction, for sure, the UVEC cells are an important model because they are of human origin. These are endothelial cell. This is an endothelial cell line isolated from the vein of the umbilical cord, and like the umbilical cord, the umbilical endothelial cells, they exhibit a cobblestone phenotype when lining a vessel walls. This is a picture of these cells, and this is also a paper, a recently published paper, which is kind of collecting most of the evidence and the uses regarding this type of cell types. They can be used to evaluate the function and the pathology of endothelial cells, including angiogenesis, neangiogenesis, also in the context of tumoral angiogenesis, endothelial cell differentiation, endothelial dysfunction, which is pivotal in atherosclerosis, and also the modulation of endothelial tube formation, because these cells are forming, tend to form endothelial tubes, which are a prototype or a precursor of blood vessels, and the formation by, and their role by anti-angiogenic agents. You can see here several biomarkers that can be utilized to trace and to identify these cells. And this is the immunofluorescence picture showing CD31, which is a peculiar marker of endothelial cells, here in green. This is called also platelet endothelial cell adhesion molecule, which is again involved into the adhesion of these cells. And in blue, of course, is a classical DAPI staining. Okay, let's move now to another cell type, monocyte macrophages. You know that monocytes from the blood flow, they start adhering to the epithelium, to the endothelium more better, because there is an expression of adhesion molecules like VCAM and ICAM, VCAM1, ICAM1. They migrate, enter beneath the endothelial lining, and then become macrophages, and they become they express phagocytic ability, especially towards LDL, oxidized LDL, and so on. And they're also able to exert a specific phagocytic activity, which is atherosclerosis, which is the ability to phagocyte apoptotic bodies, to clear the area. Coming to cell models, I think that the most important one here is the human THP1 cell line. These are monocytes isolated from peripheral blood from an acute monocytic leukemia patient. For the few of you who are not familiar with the cell lines, usually they are derived either from human tumors, I mean the human cell lines from human tumors, or they are immortalized by viral infection, SV4, TT antigen, or other types of viral immortalization. So these cells they have a peculiarity. They are cultured in suspension, so floating cells are viable. Here, this is a picture of THP1 cells, and you can see here that THP1 monocytes, these are the ones that you actually buy, can be differentiated with phospholester towards a macrophage phenotype, and further with a different cocktail of which includes phospholester, interferon gamma, and LPS. You can kind of derive from them a polarized macrophage, M1 polarized macrophage. If you want to obtain an M2 macrophage, you see here 48 hours with a different cocktail for phospholester, IL-4, and IL-13. So this is a very, very useful model and can be used in several ways. One typical way is the fact that you stimulate with LPS, they produce cytokines, pro-inflammatory cytokines, and then you may intervene with a drug or a phytocomplex, for example, in order to try to inhibit the release of pro-inflammatory cytokines. This is a paper, not so recently, if you want, published in atherosclerosis 10 years ago almost, showing that the use of THP1 cells as a model for mimicking the function and regulation of monocytes and macrophages in the vasculature. One main pro-inflammatory event that is shown present in macrophages is the activation of the NLRP3 inflammasome. So you know that this activation leads to activation of caspases which cleave pro-IL-1 beta to IL interleukin-1 beta, which is per se pro-inflammatory at the level of the vessels, and also it is inducing IL-6 production by the liver. So there is a convergence of these two very important pro-inflammatory cytokines. Now, this is a paper which is interesting because it says that inhibition of NLRP3 inflammasome attenuates foam cells formation by THP1 macrophages by suppressing oxidized LDL uptake and promoting cholesterol efflux. So again, inhibition on NLRP3 in this model is something that is interesting because probably is one of the events able to, let's say, reduce cholesterol accumulation in the plaque, at least at the experimental level. Let's now move to smooth muscle cells. As I said before, they change the phenotype. They migrate from the media to the intima. This activity is promoted by IL-1, TNF-alpha, and also by PDGF and TGF-beta. So it's a complex, let's say, set of triggers. And if you want, the phenotypic switching of these cells is very, very complex, in the sense that you can see here that authors, this is a very recent paper, a review paper, authors actually were able to describe at least 10 or more different phenotypes derived from these vascular smooth muscle cells, which means that probably different plaques may have a set of different smooth muscle cells phenotypes and change it towards a different phenotypes. And this is maybe in part also explaining the differences in the different plaques. You know better than me that the atherosclerotic plaque here is not only an accumulation of macrophages with lipids inside the foam cells, but also there is something that is a fibrous cap here, up here, which is something that is more or less present. And apparently this is produced by several cell types, especially by changed or modified smooth muscle cells. Clearly, apparently, or probably, not all muscle cells present in all plaques are able to produce a good fibrous cap, which is able to stabilize the plaque. But maybe if we have different cell types, different, let's say, phenotypes from smooth muscle cells, we can have myofibroblasts-like, osteoblasts-like with calcification, and so on. So these changes may explain the variability and the different details of the different plaques. I'm not going into the list of these markers. Of course, if you look at osteoblast-like differentiation, you can find some osteoblast biomarkers like RANKS2, for example. Then you have macrophage-like phenotype. They can also become foam cells somehow. So this is interesting. And they have as surface markers, for example, CD36, and so on. So I'm not going to the details, but it's very important to go into this cell-specific evaluation. This is another very complex picture showing that several triggers here, not only cholesterol, but several others, may ultimately cause a different phenotype. So the triggers promoting somehow inflammation, NLRP3 activation, and so on, may ultimately, within a specific plaque, within a specific organ or patient, may produce different phenotypes. A very complex issue. This has been kind of managed utilizing different cell types here, cell lines, SM22 alpha and others. These are from mice, and also we also have some human cell lines. And according to the differentiation medium you see here, you can obtain over 48 or 72 hours or a longer time, 14 days, one of these phenotypes, osteoblast-like, fibroblast-like, macrophage-like, and so on. And this is going to clearly be associated with specific biomarkers and clearly biological activity. Let's move to the origin of most of the lipoproteins and lipids in the circulation, hepatocytes. I don't have to tell you how lipids are produced and metabolized by in the liver, by the hepatocytes. You can see here many of the players, the LDL receptors. You can see here also, rather recently, identified players like PCSK9. So this is a very complex biology occurring in the liver. So also liver cells can be part of the process within the context of the human body. Another point which is important and which is involving the liver is the liver steatosis, or in this case is the non-alcoholic fatty liver disease, or also nowadays called metabolic associated fatty liver disease, which is a disease absolutely frequent in people with several features promoting cardiovascular disease, atherosclerotic cardiovascular disease. It is a metabolic syndrome with essential obesity, type 2 diabetes, hypertension, dyslipidemia, and chronic kidney failure. Now, putting together all these things, the direction of the relationship between fatty liver and cardiovascular disease is bidirectional. So far, so it is very important, very interesting to deal with human hepatic cell lines. You can see here a rather long list, at least six human models, cell lines of hepatocytes. I tell you now that the most popular is the HBG2 cell line. I will go quickly over the other ones and show you a few data about HBG2. Okay, primary human hepatocytes are ideal because they can be resected from liver tissue, but as you know, like all primary cell cultures, they are not lasting too long, they are not very practical, their viability is strongly influenced by the surgical and transport conditions. There is a genetic variability between donors clearly, and of course there is a phenotypic instability. So interesting for some studies, but not absolutely practical. The HUH7 is a model with used especially to study hepatoma, that is hepatocarcinoma, and hepatitis C infection. So interesting, but not that useful maybe for us. Like the HEP3B cell line, this has the hepatitis B virus integrated into the genome. So interesting for this specific topic. Like the HEPA-RG, this is also studied for hepatocarcinoma and hepatitis C infection. A good model for enzyme induction, but several limitations, including a wide batch-to-batch variability. Then there are the hepatocyte-like H cells, HLC. These are similar to primary hepatocytes, but they don't have the limitations in culture. So anyway, they are collected from human stem cells. So if you want, here there are some ethical limitations in their use. So eventually, we come to the HBG2 cell line, which is very popular. I think there are more than thousands and thousands of papers utilizing this cell line. Although, and this cell line is interesting for several reasons, especially for our purposes. Now, we have pros and cons. The list of pros, I don't know, this is too longer than the cons, not because we are working with this, but actually there are some nice features. High availability, stable phenotype, easy to manipulate, highly proliferative. They are quite robust and easy, quote unquote, to culture. Although, you know, experimentally, all of us always experience problems with everything, but this is part of our job. And then what is interesting for us is now shown here in green. For example, the expression of various metabolic functions of the liver linked to the metabolism of cholesterol and triglycerides and lipoproteins. They express LDL receptors. They internalize and do metabolism of chylomicrons and lipoproteins. And also, there is a whole set of apolipoproteins and lipids which are secreted. So, for our purpose, they seem to be quite good. There is a poor expression of key enzymes linked to drug metabolism, so maybe they are not the best model if you want to study a liver metabolism of drugs. Also, there is a poor expression of protein involved in the metabolism of xenobiotics. So, toxicologically speaking, maybe this is not the best model. But anyway, we are interested in atherosclerosis, so this is probably is a good model. As I told you, healthy liver can become steatotic, and this is the non-alcoholic fatty liver disease. And under certain conditions, because of some genetic predisposition and other factors, the disease may go on towards steatohepatitis, which is fibrosis, inflammation, and uncontrolled proliferation, which may end up to cirrhosis and even hepatocarcinoma. You can see here a picture in our lab, collected in our lab. This is the oleic, the staining with the oil red ore. And so the reddish color indicates the presence of triglycerides. This is the normal cell line. This is these are cells treated with oleic acid, 100 micromolar for 7 days. This is the paradigm of treatment to obtain a quote-unquote steatotic-like model in vitro. Okay? And these are some of the features that are associated with this paradigm. And this is associated not only to increase of triglycerides, but also to increase production and secretion of TNF-alpha, so pro-inflammatory pattern, okay, increase of GLUT2 expression, and some other features. These are some data we obtained. Glucose uptake is reduced, so cells become, let's say, more, let's say, insulin resistant. The triglyceride content is not only visually increased, but also the quantitation tells us that it is increased. Oxidative stress is also increased. So some of the features of a dysfunctional hepatocyte. Let's move to the last part of my presentation. And all of us know that a major player in atherosclerotic cardiovascular disease is the presence of adipose tissue, not only here in the belly, the visceral adipose tissue, but also in the epicardial area and, let's say, along the big arteries. So we have healthy adipose tissue. We can have hypertrophic dysfunctional adipose tissue, which is also infiltrated by macrophages, activated macrophages, with production of pro-inflammatory cytokines and CRP, and producing high levels of free fatty acid and lipotoxicity. This is a systemic effect of adipose tissue, but also nowadays we can talk about a paracrine effect. Indeed, this is a blood vessel, normal artery. You can see here that there is a perivascular adipose tissue, and like many compartments with physiological function, many adipose compartments with the physiological function, you can see here the production of proper amounts of adiponectin, which is anti-atherosclerotic, nitric oxide, IL-10, and so on. And this is the normal blood wall, vessel wall. But if you have a dysfunctional switch of these adipocytes, you also have a metabolic switch, and the pattern is different. So more leptin, visfatin, FABP, more production and release of pro-inflammatory adipokines and also cytokines, and these are going to affect the nearby tissue. So basically, now we can believe that the atherosclerotic plaque is due to damage from inside, from the blood flow with monocytes, lipids, and so on, but also with some pro-inflammatory damage and oxidative damage from outside. This is actually a bidirectional work, according to some authors, in the sense that the adipose tissue can affect negatively not only muscle cells, but also the endothelium, and vice versa. The endothelium, the dysfunctional endothelium may affect not only smooth muscle cells, but also adipocytes. We have several cell models of adipocytes of murine origin, the 33L1 and the F442A and the OP9, and the SW872, which is a cell line of human origin. Now, these are mostly, let's say, immature cells. Actually, if you want, the 33L1 are fibroblasts, which can be transformed, differentiated to pre-adipocytes via a treatment with a cocktail with insulin, IBMX, and dexamethasone. Instead, SW872 cells are human liposarcoma cells, so they are derived from a human liposarcoma, a male 36-year-old, and they have a fibroblastic morphology. They are pre-adipocyte-like, so they are here during the maturation pipeline. They have 80 chromosomes, but of course, we always deal with an increased number of chromosomes in these cell lines when they are of tumoral origin. They grow in monolayer culture. Now, also these cells can be pushed to change their phenotype in order to go from a pre-adipocytes, let's say, or less dysfunctional or quote-unquote normal, let's say, adipocytic phenotype towards a young adipocyte and mature adipocyte profile. We have two ways to reach this level here, this mature adipocyte pattern. One is the oleic acid. Oleic acid, as you know, is a ligand and activator of PPAR gamma, so it is a very strong type of intervention. Again, 7 days for 100 micromolar. But these cells, since they are pre-adipocytes, they are committed to become, over time, to become mature adipocytes. This spontaneous differentiation takes 17 days. So we compared the two different types of differentiation, and I'm showing few data. First of all, what happened is that, again, like in FG2 cells, oleic acid increases the amount of triglycerides in the cells. This is shown here with Euler-Redoux staining, as well as with triglyceride content measurement. Is basically double, almost double. In parallel with that, cells are more glucose resistant in the sense that the glucose uptake is reduced and interleukin-6 release is increased. So this is a nice, in a way, nice, experimentally speaking, a nice dysfunctional pattern. The dysfunctional pattern can also be underlined by some gene expression studies. The oleic acid treatment is not changing CBP alpha expression. It is not changing PPAR gamma expression, but it is reducing uncoupling protein 1 expression here. So basically, we are running away from a white, maybe, I don't say brown, but brownish maybe, or functional adipocyte, and we are going towards a true, probably true dysfunctional phenotype. When we go towards when we push the cells to spontaneous differentiation, 17 days, you see there are no changes in UCP1 expression, but there is a change in PPAR gamma expression and CBP alpha. Actually, all of us know that normal differentiation of adipocytes is associated with increase of CBP alpha and PPAR gamma. And so this is a more physiological, let's say, pattern without any particular dysfunctional feature. So this model can be used as either normal adipocyte or dysfunctional adipocyte, theoretically. We are still in the process of characterization of the model. Now, we are in the era of single cell studies, which are very complex. This is a very recent paper that came out in atherosclerosis. And you can see here that single cell studies are important because we have quite important, quite high intra-plaque, intra-individual heterogeneity in cardiovascular, in the pattern of the cells. For example, smooth muscle cells, different phenotypes, maybe in the same plaque. And then immune cell heterogeneity, atherosclerosis specific antigens, different inflammatory mediators, genetic alterations, altered metabolic states, diabetes or not, oxidative stress or not, and so on, and different lipid species. So this is one point that we need to face. We are talking about cell models. So our cell models is replicating which type of the multiple cells present in the very same plaque. Now let's move to the last part of my conversation, just showing you some of the present/future approaches to cell models. The 3D systems, organoids, milli and microfluidic systems. Clearly, most of us use two-dimension in vitro culture systems, using petri dishes, using flasks, using multi-well plates. These are static cultures. We can use one cell type, monoculture, or co-cultures. They can be co-cultures together or in a separating them via a transwell, so the contact is only through soluble factors. They can be in monolayer, they can be adherent, or like THP1, they can be in suspension. Okay, quite cheap, let's say, or not expensive. Research is never cheap, but not so expensive. These cells can be used for different purposes, but they are static and they are really they are not depicting or replicating the complexity of a true organ. So to this purpose, there are some models which are more complex, and they try to replicate some of the features, as you know, of normal organs. These are the 3D in vitro culture systems, the scaffold-free, the scaffold-based. So the scaffold-free are basically spheroids or organoids with the cells of one or more than one types, which are put together to form these spheroidal organs, small organs. Then we have a scaffold-based system, which can be from hydrogels or by bioprinting. And these are basically cells are put adherent to a scaffold. Maybe it can be a natural scaffold, which is decellularized. And then we put the cells in there, or with bioprinting. And then we have chip-based systems with some small, communications, like small channels here, able to communicate. And they can be multicellular cultures, and they can be also dynamic in the sense that you can have a pump, and so you have a circulation of fluid. So the 3D in vitro models, again, can be static, can be dynamic, can be used for to understand some molecular basis of tissue function. The signaling pathways. You can sample the fluid from some of them. Also, the drug responsiveness in disease states. Also, they ensure compliance with the 3R principles, which are reduction, refinement, and replacement of animal use. Now, clearly, some of them need to be better set up, but this is a very interesting model. For example, we can utilize endothelial cells or macrophages and see whether the products of hepatocytes or adipocytes may influence their biology. Again, this is repeating the concept, organ-on-chip microfluidic systems. rely on interconnections through connecting different cell types. And also, if the system is not a microfluidic, millifluidic, but a millifluidic, in this sense, we can have a larger amount of fluid that can be used for measurements. I don't want to bother you with many details, but you can see here, for example, this is an artificial bioprint of a carotid artery. Here is the series of cell types layered. You see on the ECM, then you have the vascular smooth muscle cells, and extracellular matrix, the UVEC to represent the endothelium, and the THP1 we spoke about to represent circulating monocytes, which may attach. And now you see here an experiment quite small, but an experiment with atorvastatin. Treatment with increasing doses of atorvastatin is able in this system to reduce the number of foam cells here underneath the UVEC cells. And this is another type of artificial small artery, and this is a model of stenotic artery. I leave you these long tables which depict the cell types and advantages and disadvantages of the different models. Clearly, we need to start from 2D cell cultures to know very well the cells, and then we may move to the other systems, maybe using the same cells, the same cell types. You see, they are listed here. So basically, the cell types or cell lines are more or less the same, but can be assembled into this type of organ-like, or let's say organ-like systems, or even connection of organs. Clearly, the biology of atherosclerosis is very complex. It's not relying on one cell type, separated from the others, but there is a quite complex relationship, which is depicted here in the human body. So in any case, I think that the use of three-dimensional systems or interconnected microfluidic systems can really, kind of replicate at least a dynamic system, which is actually what happens in vivo. So I thank you very much. I'm ready to take questions.
Thank you very much, Paolo, for your excellent presentation. Let's see if we can have some questions from the audience. Johannes wants to ask a question, if I can see well. If somebody wants to ask a question, yes, Johannes, please. She cannot unmute. Okay, Miron, are you online? I think Miron is the host. I don't know if he can do it.
I think it works. Thank you very much. Can you hear me?
Okay, yes, it works, yes.
Okay, good. Thank you very much for this interesting presentation, Paolo. I have a question concerning the 3D models. We had actually, I think it was a Nature Medicine paper by the Penninger group. I think Weismann was the first author claiming an vascular organoid model, but in my view, I mean, it looked good, but in my view, one of the problems was that, of course, you do not have a perfusion in such a, even if it's a sophisticated 3D organoid with endothelial cells and smooth muscle cells and so on, you don't have any perfusion, you don't have any blood circulation. So what is your opinion on that? I mean, it was a highly kind of appreciated paper, but I think it was still quite artificial, actually, the model.
Yeah, actually, first of all, I don't have personal experience, but reading the papers, they also can model and modulate shear stress to the wall. So they're trying to, for example, all of us know that atherosclerotic plaques are present whenever you have a high shear stress, a stress of the wall, bifurcation, and so on. So there are some models in which they can modulate, increase, for example, shear stress of the wall. So this is something that is somehow replicating some of the things. Clearly, let's not forget that these are models. Now, the thing is that we get close, we cannot replicate a human being, also because clearly the variability, the genomic variability and the lifestyle or whatever of human beings is unique to each of us. I think that it's a matter of assessing the value, the cost effectiveness of these things, okay? Because clearly technology can be fashionable or can be also useful. So it's important for us to understand what is actually cost effective and telling us the true information. So this is what I say. Anyway, clearly there is, I mean, blood is carrying not only lipids, but it's carrying so many things. So for example, we are all of us dealing more or less with small non-coding RNAs. So again, so there are so many features that should be put into the model.
Yeah, sure. Thanks.
Okay, any other questions? So, Paolo, can I ask you something about HIPG2 cells? Because we have been using HIPG2 cells for decades. So these are old cells. So do you regularly check the phenotype of these cells? I mean, if they are indeed the hepatocytes by, let's say, by measuring the expression of some hepatic markers, or you just take it for granted that these are hepatocytes and you just do the experiments?
Okay. Theoretically, everybody should check all these things. You can trace back the FG2 cells back to the 1970s, right?
Yes.
1973, I guess.
Mm-hmm.
So clearly, personally, maybe if you are using these cells, right?
Yes.
Okay, so I think that one of the goals that we can have in our project is to select a set of biomarkers and altogether and do a check in our cells. This is a good point, because we check by eye the phenotype, the morphology, but we are not really going into the details. For example, they have something like 55 chromosomes, right?
Yes.
Yeah. So, I mean, there are some features that should be checked. And I think that this is a very good point. So, because the quality of the cell models that you have. So also, well, luckily they are from a cancer, but not from immortalized cells, because I have experience with immortalized neurons, generation secreting neurons from mice, and with the SV40T antigen, and the researcher who did them told us, well, you know, after the 30th passage, they get crazy. They can lose. I have, when I was even the second year of my PhD program, I was so sorry. I did so many experiments with these cells, but they lost a receptor for neuropeptide Y. I was looking for activity, and they were not responding, simply because they had no receptor anymore.
Mm-hmm.
So this is a very good point. This can be a small, I don't say project, but a small quality control that we can do.
Mm-hmm.
A sort of SOP. This is a standard operating procedure, like when you collect samples from humans and store them and centrifuge them, whatever. We need to introduce these SOPs also and the first candidate are going to be these cells.
Yes. So another issue with these cells is that some of the, let's say, hepatic transcription factors are overexpressed. I mean, their expression is exaggerated. And this sometimes creates some problems that, especially if you do gene regulation studies, you cannot see any differences in expression of target genes. But let's say, with primary hepatocytes, the responses are much better. And now, since we're talking about the primary hepatocytes, of course, you said that they are not so, let's say, convenient because they have limited lifespan, etc. But it's very important. They are very important when let's say you want to study what's happening in the liver of certain mouse strains. Okay, these are knockout strains. These are very important. I mean, this to isolate primary hepatocytes instead of, let's say, knocking out the endogenous gene in HepC2 cells, you just isolate primary hepatocytes, let's say, from the knockout of PPAR gamma or from MXR, right?
Yes.
So
This is a good point. This is a good point. And now I have to add that, for example, this model with the oleic acid is a very extreme model because it's very powerful. I didn't show you some data with let's say natural compounds. We have some project on agro-food waste, extracts from agro-food waste in order to develop let's say nutraceuticals for use in the metabolic syndrome, dyslipidemia, and so on. And clearly, some of them are effective in reducing triglycerides, but it takes 48 hours or more, in the sense that the oleic acid is a very extreme and powerful model, the one with this treatment.
So you have two questions from the audience. One is from Fabio. I know it is very difficult or impossible to predict future, but are we going to still use 2D cell culture systems in 5 to 10 years time?
Sorry, the last word. I didn't get it.
In 5 to 10 years time, are we going to still use?
Who
2D
knows?
cell cultures?
Who knows? Who knows?
So
you know what? Now
future is uncertain.
No, I mean, one thing that is nice, if you have to test drugs, or I mean, we have this project testing some natural compounds. Okay, so different doses, different concentration, different time courses. So this is just a test. So you can test if they improve glucose uptake, if they improve inflammation, and so on. Clearly, you don't know if this is going to be an action that is going to take place in humans, in the human taking this type of products. But at least you can screen these things. You can screen these activities. We did it last year in 3 different cell types, including also the CACO2 cells, which are the intestinal cells. So clearly, they are very, very much, models, reductionist models. So you cannot expect too much, I think.
And another question from Bianca Descal. Hi, Paolo. I would like to know if you can combine hepatocytes and adipocyte cultures and differentiate them together with oleic acid.
Yeah, this is a very good point. Now, we will try. We are trying to now to select to decide whether it is better to have cells which are in contact or which are in contact, not in physical contact, but with the transwell, and so there is only the soluble substances can affect, or using the conditioned medium from one cell type to the other. Okay, we have some preliminary experiments with smooth muscle cells that apparently are affected by the conditioned medium by hepatocytes or adipocytes. So yes, it's a matter of putting together. This can be important.
Okay. Any other questions? Anybody wants to ask a question and open the microphone? Miron said that he can unmute you if you want. So there are no further questions, Paolo. So again, thank you very much for your excellent presentation and discussion.
Thank you very much. So I can close just saying that these are nice models, are single cells. So let's not, when we write a paper, we cannot really speculate too much. Okay, we can just describe and, no speculations at all, if you want. Okay, thank you very much to everybody for your attention and for your time.
Thank you very much, Paolo. Miron, are you still online? Or he's not? He's not online, probably. Okay. So we can close the webinar here. Thank you very much. I believe the next webinar will be next month, and
Yes.
will be about animal models.
Great,
of
fantastic.
atherosclerosis.
Okay,
Yes.
wonderful.
Okay, so
Thank
see
you
you
very
in
much.
the next webinar.
See
Thank you
you
very
next.
much, everybody. Thank you very much for attending. See you in the next webinar. Bye.
Thank you. Bye-bye. Bye-bye.
Bye-bye.